Welcome students. This is for 4th semester student Pharmacognosy and Phytochemistry 1. We are going to see the last chapter in your syllabus. It's very important chapter. I have seen that more than 40% of questions have been asked from this chapter. I have told another chapter also. This chapter I have seen most of the time they are asking 5 mark question and 2 mark question. Minimum 6 to 7 questions have been asked from this chapter. That is chapter number 5, unit 5 is unit study of biological source, chemical constituent and uses of drugs and natural origin containing following plants, especially drug products, fibers in which cotton, jute, hemp and hallucinogen they gave tobacco and as well as cannabis. Cannabis and hemp, it comes even in fibers also. Teratogen, they have given only 2 and we should know what is teratogen and colchicum veratrum. And natural allergens, you should know what is allergens and classification, preparation and standardization of allergenic extract. This also they have asked questions. And primary metabolites, they have given general introduction about primary metabolites, how I have given in secondary metabolites. Detailed study with respect of chemical constituent sources, preparation, evaluation, preservation, storage, therapeutic uses and commercial utility as pharmaceutical aids and or medicines for the following primary metabolites specifically selected, like carbohydrates in which acacia, agar, starch, tragagan, honey, and they are given in proteins and enzymes, gelatin, casein, proteolytic enzymes like papain, bromelain, serratiopeptides and urokinase and streptokinase, pepsin. Lipids, waxes, fats, fixed oil in which castor oil, chalmogra oil, wool fat, beeswax only. And marine drugs, novel medicinal agents from marine sources, especially antiviral, antimicrobial, anti-cancer and cardiovascular agents obtained from marine drugs. So it's an interesting chapter. They have asked many questions. So it's a 5th chapter for the 4th semester B Pharm students, Sree Krishna College of Pharmacy and Research Center, Parassala. And kindly, any doubts, you can mail me and you can contact me in my WhatsApp number. You see, I am going to discuss about only 4 years. They have conducted exam in Kuhas since 2019. 2020 we got a break due to Corona, and we had 2021 and 2022 and 2023 and 2024. So you see, we have taken only 4 years question since 2019. So in which you can see I have had more than 75 questions they have asked within this year. So it's an interesting to know that in one question paper they are asking more than 7 questions. See, this is a 5 mark question which is asked, write short notes on cotton from fibers, and plant allergen 5 mark question in July 2019. and describe the biological source, preparation, chemical constituents and uses of wool fat, 5-mark question in July 2019. Describe the biological source and uses of honey, 2-mark question in 2019. And different sources of starch, 2-mark question in July 2019. Describe the source and therapeutic uses of urokinase, 2-mark question, July 2019. Name any 4 antimicrobial drugs from marine sources, 2-mark question, July 2019. Write the pharmacognosy of cotton, 5-mark question in December 2019. You just see 7 questions I have asked in the July 2019 question paper, and in December 2019 cotton again they have asked. Chemical test and identification of acacia, 5-mark question in December 2019. Biological source and chemical constituents of tobacco, 2-mark question in December 2019. And the method of preparation of agar, 2-mark question in same question paper, December 2019. And source of pharmaceutical uses of beeswax, 2-mark question in December 2019. Name any 4 antiviral drugs for marine sources, 2-mark question, December 2019. And then comes April 2021. 2020 we had a break due to corona. Define lipids. Write a note on source, methods of production, constituents, tests, uses of castor oil. 10-mark question they have asked in April 2021. And discuss on source and method of collection of wool fat, 5-mark question they have asked 2021. Preparation and standardization of allergenic extract, 5-mark question, April 2021. Method of preparation and constituents of cotton, 5-mark question, April 2021. Differentiate acacia from agar by chemical test, 2-mark question, April 2021. Mention the source and uses of bromelain, 2-mark question in April 2021. Honey, 5-mark question in December 2021. You see, in April 2021 also they have asked 10-mark question, 5-mark question and 2-mark question. You see that it's more than, usually they are asking more than 22, 25 marks. Even some question they have asked 27 marks also they have asked. So you just see in 70-mark question they are asking more than 40 to 40% coming. If it is 30 marks they have asked in one chapter. So it's an interesting chapter which you have to see all the questions, and you have to see according to the syllabus and according to the lecture. You prepare the notes and find out if anything is missing. You have to ask with the teacher for the guidance to study from which source to take. Chemical test for acacia, 2-mark question in December 2021. Write the biological source, chemical constituent and uses of cannabis, 2-mark question in December 2021. Define teratogen with example, 2-mark question in December 2021. Mention the source, constituents and uses of chalmogra oil, 2-mark question in December 2021. Give the source of pepsin and casein, 2-mark question in December 2021. What are primary metabolites? Give examples, 2-mark question in December 2021. What are hallucination or hallucinogens? What are hallucinogens? Add a note on cannabis as a hallucinogen. So you should know about hallucinogen means what, and then about cannabis, how it acts as a hallucinogen. 5 mark question in June 2022. And write the source, method, collection and constituents of Trivandrum. 5 mark question June 2022. It is an unorganized drug. It's a gummy exudate from astragalus gummifer. You should write how it is collected and you should know about the constituents. 5 mark question. And discuss marine drugs as a source of antiviral and examples for 5 mark question in June 2022. You see the 3 5 mark question they have asked and still 2 mark questions also they have asked. Mention the source, active, constituent, uses of gelatin. 2 mark question in the same question paper June 2022. And in the same question paper you can see give the source, family and constituents of plant fibers. 2 mark question in June 2022. Even in the June 2022 again 2 mark question. What are the usual adulterants of honey and how it is tested? 2 mark question June 2022. And confirmatory test for castor oil. 2 mark question June 2022. And you can see the next November 2022 the year question. Describe the biological source, chemical constituents and uses of cannabis and colchicum. 5 mark question November 2022. Cannabis is used for, you can see, hallucination. It is an hallucinogens. It causes hallucination. And colchicum is a teratogen agent which is given in the syllabus. So 5 mark question the 2 drugs they have asked in November 2022. In write the source and method of preparation of castor oil. 5 mark question in November 2022. Biological source and use of jute. 2 mark question in November 2022. The same question paper. Feast test for honey. They have asked chemical test. 2 mark question in November 2022. Source and therapeutic use of chalmogra oil. 2 mark question in November 2022. Name any 4 anti-cancer drugs from marine source. So 2 mark question. You should write the name and as well as the structure, chemical structure. And question 2 mark question in November 2022. Explain about biological source, chemical constituent and uses of veratrum is a teratogen and tobacco as a hallucinogen. And 5 mark question in March 2023. And write the source, chemical constituent, chemical constituents and as well as chemical test and uses of gelatin. 5 mark question in March 2022. Describe or discuss the marine drugs as a source for cardiovascular agent with examples of 5 mark question in March 2023. Write a brief note on the source of therapeutic use of castor oil. 2 mark question in March 2023. Write a brief note on source and therapeutic use of papain, the enzyme papain. 2 mark question in March 2023. What are teratogen? Give the source, constituents and uses of colchicum. 5 mark question in July 2023. Cardiovascular agent of marine sources. 5 mark question in July 2023. Source and uses of streptokinase is an enzyme which is responsible for various lysis of clots and urokinase. 5 mark question in July 2023. Differences between primary metabolites and secondary metabolites. 2 mark question July 2023. Give the source, constituents and uses of agar. 2 mark question July 2023. Source and uses of enzyme from animal sources. What enzymes which is obtained from animal sources. 2 mark question July 2023. And you see 51 questions again. It is 75 I told. 51 question July 2023 itself. 2 mark question. Source, constituent and uses of castor oil. Source constituent uses of cotton and jute, 5 mark question. It is in February 2024. What are hallucinogens? And write a note of any one source of drug. You can write cannabis, you can write tobacco, such drugs you can explain, any one you can write. 5 mark question in February 2024. And write note on proteolytic enzyme. You should write a proteolytic enzyme papain. You can explain. 5 mark question of February 2024. And standardization of allergenic extract, 2 mark question in February 2024. Source constituents and uses of acacia, 2 mark question in February 2024. Explain chalmogra oil, 2 mark question, February 2024. Chemical test for gelatin, 2 mark question, February 2024. Anti-cancer drug obtained from marine source, 2 mark question, February 2024. Explain about biological sources, chemical constituent and uses of cotton and hemp, 5 mark question in May 2024. Write a short note on standardization method of allergenic extract, 5 mark question, May 2024. Write a short note on source, chemical constituent and evaluation test of honey, 5 mark question, May 2024. Discuss marine drugs as a source of antimicrobial with examples, 5 mark question, May 2024. And what are teratogens, 2 mark question, May 2024. You see how much questions they have asked in same question paper, May 2024. Define enzymes with example, 2 mark question, 2024. 3 5 mark question and 2 mark questions also they have asked in the same question paper. You see, write a note on biological source and uses of cannabis, 2 mark question in same question paper, May 2024. And the same question paper, write a note on carbohydrate, 2 mark question in May 2024. And again in the same question paper, May 2024, you can ask, they have asked, write a note on gelatin, 2 mark question of May 2024. You just see that 3 5 mark question and 5 2 mark question, 10 and 15, 25 marks they have asked in one question paper. Minimum 20, 25, 27 and source and constituents and uses of starch in honey in 5 mark question in the next in December 2024 recently. And define what all allergens with example, 2 mark question in December 2024. And a preparation of medicinal castor oil, 2 mark question, how to prepare from the seeds of Ricinus communis, castor oil, how it is prepared, December 2024. And differentiate between proteins and enzymes, what all the difference, which example, with examples, 2 mark question in December 2024. Colchicine is a teratogen and it give the source and uses, its uses, 5 mark question as in December 2024. So these all are some of the questions which I have listed out. Write down and make sure you can answer all this question when you are when I am completing this unit, or when you study this unit. Initially itself you should know what all the questions they have asked from my syllabus according to Kuhas. It is needed for the students to know about the basic thing of a syllabus and the questions which is asked previous year. So 4th semester student, unit 5 is being a very important, more than 45% of questions repeatedly they are asking in each question paper. So you have to go through is very clearly according to the syllabus, and the lecture you prepare the listening the lecture and write the assignments as your notes and submit without fail. Most of the students are doing it and some of the students are lacking in it. Especially I request the boys, and especially why you are not writing, I don't know. Why you are not attending the class, I don't know. Why you are being absent, I don't know. This is not a good sign of being absent and being not attentive to the class and being respecting the teacher, for obeying the teacher, and discipline is main important for your academic success. So kindly my request for the students, write down and submit your notes regularly to my office. I expect this lecture, the coming lecture with the questions, write down all these questions and write down this one small lecture which I am giving for fibers: cotton, jute and hemp. Only 3 units, 3 topics which we are discussing today, because we have spent some time for reading the question paper and the syllabus. So you see here in fibers, fiber means you may be knowing about many fibers like cotton and jute, wool. These all you might have seen, but we are going to see about the fibers which they are given in the list, about the fibers which we are going to see about in this pharmacognosy and phytochemistry 1. And here, study of biological source, chemical constituent, uses of drugs, natural origin containing plant drug products. But even they are asking about the wool fat, but anyhow this 2 times they have asked, so I will give you that detail also. But here fibers in natural sources, cotton, jute and hemp I am discussing. Hallucinogen is also there, tobacco and cannabis. Teratogen also, colchicum and veratrum according to the syllabus. But in the question paper they are asking in about wool fats also, and the wool, how they will produce, that is also we can see in the topic I used to discuss about that. But cotton, jute, hemp we are going to discuss now in this lecture. Natural fibers can be classified according to the origin. So how they are classified? Sometimes they may ask in natural fibers detail also, 5-mark question. So the students should know how they are classified. Even the classification is done by vegetable, that means the plant source, or cellulose based fiber 1, and the animal or protein based fiber, and you can see regenerated and synthetic fibers, and fibers in mineral classes. So like that 4 classes we can differentiate. In the number 1, vegetable or cellulose based classes, including such important fibers like cotton, flax and jute. And when you come to animal and protein based fibers, include wool, mohair and silk. When you come to the third one, regenerated and synthetic fibers, include nylon, terlin, orlon, viscose, alginate fibers, etc. And then when you come to an important fiber in mineral class, it is asbestos. So the student should have and categorize how the fibers can be classified: plant source, an animal source, and regenerated and a mineral source. Like that you should have a classification. And yes, I told 1, 2, 3. Remember, vegetable or cellulose based fibers, animal or protein based fibers, regenerated and synthetic fibers, fibers in the mineral class. Examples are very important for the students to remember. And the vegetable fibers can be divided into smaller groups, like based on their origin, or origin from where they are getting origin within the plant. So within the plant, where it is originated, where it is obtained. The fibers originated as hair borne on the seeds. Above the seeds will be covered with the trichomes, or inner walls of the fruits. Like a fruits, there will be inside the cotton will be there, where each fiber consists of a single long narrow cell. So fruit walls also, like even coconut, you can see this one is an hair trichomes in the seed. So that is cotton, and this is kapak, which is like a fruit, big tree. In Malayalam we will say elevencham, that means it's a big tree where we can see the cotton inside the fruit. And you can see this is coconut shell and coir. These all comes in the first category. Fibers originated as hair borne on the seeds or inner walls of the fruits. So this is the three examples. The second one, you can see blast fibers occurring in the inner blast tissue of certain plant's stem and made up of overlapping cells. Like you can see hemp, the stem part will be taken and the fibers can be prepared. And even see acacia, that means like in the banana plant, different types are there, and there you can see acacia is also prepared from that fiber. Ramsey fiber from the plant stem part, and this one is flakes, the linseed stem. Flakes also, they are also stem part will be used as a fiber dried. And jute fibers, and you can see this one, henequin is not, looks like an aloe, but it is not aloe. It is a stem part will be taken, and you can see that will be used for preparing fibers, and it will be dried and you can use for various purposes. So you should know that this is the second part. Blast fibers occurring in the inner blast tissue of certain plant stem are made up of overlapping cells. And here the third one is fiber occurring as a part of the fibrous vascular system of the leaf. So you can see this is sessile fiber. Sessile fibers are same, it is not aloe, it is also like hemkenquin, like it is cells and fibers, is also a plant. The leaf part will give you the fiber sessile. So sessile fibers are, you can see, occurring the part of fibrous vascular system of the leaves. So there are three types of category which can be done. Number one, first we are going to deal with, according to the syllabus, cotton. Cotton, you can see this is the plant, the cotton plant, botanical source. Cotton consists of epidermal trichome as shown in the picture, trichomes of the seeds of Gossypium herbaceum linn and other species of Gossypium of the family Malvaceae. The balls of cotton, you can see the balls of the cotton in the picture very clearly for you to know. And the preparation of raw cotton, the balls of cotton are collected from the ripe and the dehisced or you can say dehisced outside when it comes from the fruit of Gossypium. When it opens, the trichomes are separated from the seed by ginning process. There is a machine using ginning machine as shown in the picture. It will remove the trichomes from the seed using the machine is called gin. And this, which the trichomes are drawn through a narrow space as shown in the machine picture, the mass of these separated trichomes constitute raw cotton. And this raw cotton thus prepared contain impurities, chiefly coloring matter may be present of about 0.6%, and of wax also, oil may also be present. This should be removed. So then only it will become an absorbable cotton. Otherwise it is non-absorbent because of presence of this wax, oil and the coloring matter, which forms a film around the fibers and rendered them as a non-absorbent. Once you remove this cotton, raw cotton, is then various treatments in combined with spinning and convert into yarn before removing this wax and the coloring or the oil. The thread which can be used for fabrics we can use as a non-absorbent cotton for the cloths. But absorbent cotton as shown in the picture, if you want to make it for surgical preparation, it need to undergo certain preparation for absorbent cotton. So absorbent cotton wool is prepared from the various cotton waste obtained during the process of raw cotton for making the yarns, that is also can be taken. The waste which is taken, the shortened the products which is taken after ginning, the waste after gin machine or ginning after doing the removed trichomes used for yarns, and the remaining will be used, the waste one, loose cotton is then boiled with for 10 to 15 minutes or 15 hours, not minutes, 15 hours. You under a pressure, you should give a pressure also with an 30 lbs, that means 30 pounds. You are giving the pressure in a dilute solution of caustic soda, sodium hydroxide, and soda ash, sodium carbonate. This treatment removes the fats, coloring matter, waxes, renders the trichomes to be absorbent. The trichomes will become absorbent. It will be absorbent cotton, and so that you can see it becomes an absorbent cotton. So this having a cotton absorbent capacity, the boiled material is then washed thoroughly with water, bleached with dilute sodium hypochlorite solution, and treated with very dilute hydrochloric acid. The bleached mass of the fibers are then dried, loosened, and stretched, and carved, and that is carded by using certain appropriate machine, by pressing it, and then it will be rolled, and it is used for surgical purpose. And this absorbent cotton, the capacity of absorbent is decided according to Indian pharmacopoeia or United States pharmacopoeia. You should have the procedure how to decide what is its absorbent cotton capacity, also decided by means of making it, weighing it, and making it to absorb water, and then again weighing, and the capacity of the absorbent cotton can also be decided as a main parameter to find out what is its absorbent capacity, like how you are doing absorbent charcoal. This is also absorbent capacity cotton can also be decided, because once you remove the fats and waxes coating which is present in the trichomes of the cotton, it becomes absorbent cotton. Very interesting procedure which can be used for our surgical cotton. So the resultant product is thin continuous film of absorbent cotton wool. You can say it is a cotton wool. You can tell when they ask about the preparation of absorbent cotton wool, you should remember about this preparation. Repeatedly they are asking. Several such films are superimposed on one another and rolled. That is why it is called cotton wool, you can say. The raw cotton contain about 90% of cellulose and a small amount of wax, fat, remains of protoplasm and ash. Absorbent cotton is often pure cellulose, and there are many products as shown in the picture. It can be prepared in various preparation uses. Cotton is used as a chief material for main surgical dressing. It is also used as a filtering medium and as an insulation material also. Insulating material. And you can see now we are coming to the jute. Jute is the plant which you can see like this, and the stem, from the stem part you can see the standard phloem fibers of the stem bark of the jute of what is the plant source, that is Quercus olitorius. Quercus olitorius, you can say olitorius or olitorius. Lynn is the scientist, and the second species also Quercus capsularis. That is Quercus or Quercus capsularis. Lynn family Tillaceae constitute jute as shown in the picture. Preparation of jute fiber. Jute plants are normally straight and they are unbranched. They are not branched, they are unbranched. They are cut from the base when the plant or flowering position and tied in small bundles and stacked and soaked in an stagnant water or stagnant, not stagnant, that is not target, stagnant water like pond, stagnant water, and about for 3 weeks it will be kept and for resetting. And the tracks which are covered with straws and water hyacinths, stagnants or hyacinths, to keep them wet always, and the floating water or keeping in the top and keeping always wet to protect from the direct sunlight, it can be kept. When the stem bark is well macerated by soaking in the water, maceration, maceration, percolation, where we will keep in the solvent for a longer period, well macerated by that is retting or retting it manually, or retting process, you can say well macerated, soaking them or in the water, you can say that is the word, you have to stagnant water, keeping for 3 weeks, that is called stagnant water, you can say soaked them, and that you can say that is water, you have to keep it in the water for a longer period, and so that you will get manually, what happened, the strands and the phloem fibers from the stem are separated manually by peeling off from the wood and washed freely from the surrounding softened tissues, and other dirts are thoroughly should be removed, clean water, and the fiber are then dried direct sunlight after this process and made into small bundles. So this jute fiber are exported in ballast, that means bales in the rolls made up of hydraulic pressure. You can see it is the plant, and this is the plant leaf, and this is what the bundles which they are kept in the water, stagnant water for 3 weeks, and then you can prepare and direct sunlight after peeling off. You can keep like this for drying direct sunlight, and so that you will get, I can say, even the bales of raw cotton picture also I have shown, bales of raw cotton pictures, sacks, and you can see you can prepare even Helian cloth which we can prepare from the jute, and you can see even padding, even wrapping the splint, also another use of jute, and this type of bags we can prepare. This is the threads can be prepared from the jute fibers. So constituents of jute fibers are composed of 53% is cellulose and 22% of hemicellulose, and contain 11% of lignin contains, and 1% of fat and waxes, and 1% of ash. Uses in pharmacy, jute is used in manufacturing of medicated towels and padding splints, which we have seen there, and for wrapping, as a filter and straining medium for soaking fluids, and other industrial uses. Jute include the manufacture of ropes, gunny bags, carpets, baking, carpet backings, and yarn for some fabrics and sacks. Many things are prepared. Now we are coming to hemp. Hemp comes in another topic also in cannabis. It is also cannabis sativa. This is hemp which is prepared from the stem part. It is hemp is pericyclic fibers obtained from cannabis sativa lin belonging to the family Cannabinaceae. This is the hemp plant which is you are using for the hallucination also. It's a narcotic cannabinol, which is tetrahydrocannabinol, which is present as an analgesic and a narcotic drug. It's actually, and you can see this cannabis sativa, which is the seed which is the flowering seeds, and cannabis comes under, even note I want to say, it comes under in fibers, and as well as it come in an hallucinogen. Some more detail added after few slides. So together, the cannabis sativa seeds are chiefly used to make hemp seed oil, which can be used for cooking lamps and uh, even see for preparing lacquer and paints also they are using. They can also be used for caged bird feed, and they provide wonderful source for nutrient for most of the animals. Flowers especially, and it is laser extend leaves, stems, and seeds also contain phyto and psycho, not phyto, psychotic chemical compound known as cannabinoids, and they are consumed for recreational, hallucination, medicinal, and spiritual purpose also, for getting concentration. When so used preparation of flowers, they are called as marijuana, and the leaves and preparation derived from resinous extract, and they are called hashish, and consumed for smoking and vaporizing and oral ingestion. And historically, tinctures, teas, and ointments have also been common preparation. In traditional medicine in India, in particularly cannabis sativa has been used as an hallucinogenic and hypnotic sedative. Hallucinogenic means something which is well-being feeling, not originally it is not well-being, and hypnotic, sedative, analgesic, and anti-inflammatory agent. Always whenever I used to teach from pharmacological terms, the student should know the definition, read and study. That will be helpful for your pharmacology subject. Anyhow, thank you for listening this class. I made it short because we discussed about the questions very detailedly. These fibers are very important. Only three we have discussed about: jute, cotton, and that hemp. So these three, hemp is also having, it is not only as a fiber, it is having many other uses also. So kindly read it and study clearly, and write your assignment and submit to my office. Now we will come to the next topic about hallucination and teratogen in the next class. Thank you.